Hey everyone, welcome back to another episode. Today we're diving into some groundbreaking news in the world of medical imaging,
Absolutely. Imagine being able to see deeper into living tissues than ever before. It's like exploring uncharted territories inside our own bodies.
right? So, scientists have just developed a new technique that lets us peer more than 700 micrometers deep into living tissues. To put that into perspective, that's over twice as deep as what we could previously achieve.
Previously, we were limited to about 300 micrometers because of something called light scattering. It's like trying to see through thick fog. Light gets bounced around by particles, making it hard to see clearly.
Exactly. But this new method uses what's called three-photon excitation at 1100 nanometers. Think of it like using a deeper bass note to penetrate through walls. The longer wavelength allows the light to go deeper with less scattering.
And here's where it gets really interesting. They used a multimode fiber with a compact fiber shaper. Picture a high-tech version of tuning a musical instrument to get just the right sound. This setup allows them to fine-tune the light for optimal deep imaging.
By doing this, they achieved the high peak power needed without sacrificing the quality of the image. It's like having a superpowered flashlight that can shine its beam deep into the tissue without blurring.
Now let's talk about NADH, short for nicotinamide adenine dinucleotide phosphate. It's a molecule found in all living cells and plays a crucial role in metabolism, basically how cells use energy.
What's amazing is that NAPH naturally fluoresces when excited by light, so we can use it to visualize metabolic processes without needing to add any dyes or labels. That's what we mean by label-free imaging.
Being able to see NAPH deeper in tissues means we can observe how cells are functioning in their natural state, even in hard-to-reach areas like the middle of a tumor or deep within the brain.
Imagine the possibilities. We could watch how cancer cells metabolize in real time, or see how immune cells respond to infections without disturbing them.
Exactly, this could revolutionize fields like cancer research, immunology, and neuroscience.
So breaking it down, traditional imaging was limited by two-photon excitation, which didn't allow us to see very deep because of light scattering.
But by moving to three-photon excitation and using a longer wavelength, they reduced scattering. It's like switching to a lower-pitched sound that can travel farther without getting muffled.
They needed high peak power to make this work, but high-power lasers are usually big and expensive.
That's where their ingenious use of the multimode fiber and fiber shaper comes into play. They customized the light's path, achieving high power in a compact and accessible way.
It's a brilliant solution. By fine-tuning the fiber, they control the light's shape and focus, much like adjusting a camera lens to get a clear picture.
And the best part is that this setup is more accessible and could be adopted by many labs without prohibitive costs.
This advancement also allows for faster imaging speeds, so we can capture dynamic processes like how cells move or interact in real time.
We're not just taking static pictures anymore, we're watching the movie of life unfold at the cellular level.
And because it's label-free, we aren't introducing anything foreign into the tissue, which means we're observing cells in their natural environment.
This could lead to better diagnostics and therapies since we're gaining a more accurate understanding of what's happening inside the body.
Absolutely. For example, in cancer treatment, knowing how cells behave deep within tumors could help us develop targeted therapies that are more effective.
And in neuroscience, we might be able to observe neural activity deep within the brain, opening new doors for understanding diseases like Alzheimer's.
Before we wrap up, let's appreciate how this technique overcomes previous barriers. By using three-photon excitation and an innovative fiber setup, they've pushed the boundaries of what's possible in imaging.
It's like they've given us a new pair of glasses that lets us see deeper into the microscopic world than ever before.
The future implications are huge, and it's exciting to think about what discoveries await us.
I can't wait to see how this technology will advance medical research and improve patient outcomes.
Well, that's all the time we have for today. Thanks for joining us on this fascinating journey into the depths of living tissues.
Thank you. It's been a pleasure exploring this groundbreaking development with you.
And thank you to all our listeners for tuning in. Stay curious, and we'll catch you next time.